Good evening to one and all present here. It's my great pleasure to introduce Professor S. Krupanidhi, who's currently heading the Department of Biotechnology at Vignan's Foundation for Science, Technology and Research. The university is located in Guntur, Andhra Pradesh, India. Responsibility of Dean Planning and Monitoring. Professor Krupanidhi has a long teaching experience. He served in Sri Sathya Sai Institute of Higher Learning from 1979 to 2012. Later, he joined Vignan's University and he's serving in his various capacities. Since the time he's in teaching service, he secured a variety of grants, notably DST and DBT, and also some grants from the Government of India. He conducted several conferences and organized FDPs, workshops, and various science camps. His fields of interest are immunotechnology, environmental science, bioinformatics, especially molecular phylogeny of gastropods. He has published several articles, notably 70 research articles in SCI, Scopus, and indexed journals. He is guiding PhD scholars and 4 students who secured PhD under his supervision. Thank you, sir, for accepting our invitation.
Okay.
Sir, there are some important technical notes to be given to the participants. So you are in mute mode, and we request all the participants again to kindly stop your video and mute your audio. Try to note down your questions, which will be addressed at the end of the session. And for any discussion in the middle of the talk, you can just put your hand using the raise hand option. Don't forget to send your feedback via the link which we have given in the slide. I would again post it in the chat box before the end of the session. Thank you very much. Sir, I would like to, sir, you can take over the talk.
So good evening to all webinar participants. Particularly, I am most thankful to Dr. Divyasri Nageswaran, co-founder of LLB. She is from the University of Cambridge. And also Dr. Mahesh Veluswamy, founder LLB, Let's Learn Biology and Bioinformatics, Central National Research Institute, France. And they took enormous interest through one of our young colleagues by name, Dr. Abraham, and he is the one who initiated the interaction between Divyasri Madam and our institution. And I am highly thankful to Divyasri Madam for promoting us to give a talk through this webinar. And in this context, I wish to say thanks to the Board of Studies members of our university, and also the administration of our university, Vignan's Foundation for Science and Technology, particularly because we started a program by name Immunoinformatics for our BTech biotechnology and informatics students and also MTech students. So though we proposed this program, our BOS members have accepted, so that we have been continuing this program for the past 4 years. Because immunoinformatics has been introduced, all of us are dwelling in the same field. So we are able to project some work now in this webinar, which has been done at our university. Apart from this, I'm also thankful to Dr. Abraham and Venkateswarlu and other colleagues who are there with me in doing this practice, Dr. Nazneen Bobby, and again, Dr. Indira Madam and others. Okay, so with this, again, the last thanks are due to Cisco WebEx. And Cisco WebEx is a platform under which we're all meeting happily on this Sunday. Okay. Now, I've given you some introduction about the immunoinformatics. That's what we are practicing at our place. Now, see, we are all now currently in this calendar year 2020. We are unknowingly facing a menace because of novel coronavirus. So this novel coronavirus is not new, is not new to the biological system. Already we are having in the virology taxonomy, coronaviridae, a family. Under this family, there are many groups: alpha coronavirus, beta coronavirus, delta coronavirus. So that's what the coronavirus is not new to the biological system. But the novelness in this coronavirus is it infected the human beings. And not only infected the human beings are facing a suffering and they are getting victimized. And it spread so fast that is almost all countries in this globe, the people are all suffering. So like this Spanish flu in 1918 was prevailing. The Spanish flu in 1918 was prevailing for nearly 18 months. So it nearly 20 million people have suffered because of the Spanish flu. Now, for the past 6 months, that is starting from the December up to now, we are facing the problem. So there is declining and again and escalating the COVID-19 cases in all the situations. Now, as far as now, 10 million people are getting infected. And again, in India, 4 lakh people are infected. And across the states, several of the states like Maharashtra, Delhi, Gujarat, Tamil Nadu are more infected compared to Andhra Pradesh. Okay, so this is primarily because the vulnerability of the people. And the vulnerability has come because we never got exposed to this because of any mild coronaviruses, so that we are not having any herd immunity and any vaccination for this. We never thought of having vaccination for this. Hitherto, starting from Edward Jenner and Louis Pasteur, we used to have heat-killed pathogens as vaccines or attenuated pathogens as vaccines. So that was in practice for a long time. Now, recently, the recombinant protein as vaccine. For that, this hepatitis B surface antigen is in practice now. Almost all biopharma companies are making this type of the recombinant that is hepatitis B surface antigen, that is Shanta Biotech is making, and Biotech is making, Serum India is making, those particularly in India. Okay, now recombinant protein vaccines further in development in scientific field is epitope-based vaccines. That is, an antigen means a protein. A protein means it will be having nearly more than 200 amino acids. 200 amino acids synthesizing and providing to the people to have vaccination means really it is costly at this affair. Epitope means is nearly about 10 amino acids, 15 amino acids. So the cost wise we can reduce, but it will be having the similar effect as that of the recombinant vaccine. That's what now the trend is towards the epitope-based vaccines to prepare. Fortunately, the bioinformatic tools are also helping us how to devise this type of the epitopes, design these epitopes through these bioinformatic tools. So now that's what our interest is. And fortunately, we are familiar with a few of these software tools, particularly our colleague by name Dr. Abraham Pillay. He has done much of the work. I am now presenting the work that has been done by him, and part of the work I did, particularly with regard to the phylogeny aspect of it, that also I'm projecting now. Okay. Now, one more, for a few of the participants who are among you all, webinar participants, there may be students and there may be naive people. For this purpose, I'm telling. Epitope. Epitope means it is something surface thing on the protein. That is, a protein is highly folded. None of the other biological molecules are as folded as that of the protein. Protein is having a characteristic foldings. Proteins are having the characteristic fold. So that fold is having a few of the residues in the protein are hidden. A few of the residues on the protein are exposed, and that exposed portions are called as epitope. So our defense system, like let us say our immunoglobulins, our T cells, our B cells, or our macrophages, they all identify the exposed portion. So that exposure portion is called epitope. So that is what we are now discussing much. Okay. So now ultimately by doing this, we are making our host immune system to have memory, against the epitopes that we are giving. So these epitopes are matching with the epitopes of coronavirus, so that in future, if at all we get exposed, our memory cells will fight against or neutralize this coronavirus, so that the infection will not progress. So that is the main purpose for which we have chosen this particular topic. And again, epitope, single epitope means there is a big chance to lose, so that we will be having multiple epitopes of the same protein, something like that, so that it will be having impact on generating immunoglobulins. So with this background, I am working in this. And one point I will let you know here, that is, we're all working with the software tools. So these software tools are developed based on the concepts that are given in immunology. Just like that, we cannot go to any software tool and work. So I will also introduce such of the concepts which made us to work with those software tools. Clear? Now, so with this brief introduction on this multi-epitope vaccine design, I'm going to cover in another 40 slides the following aspects. The first one is a few facts about coronavirus, because we cannot abruptly start sequence-based identification. Okay. Afterwards, one slide on immunoinformatic tools. The next one is sequence-based identification of epitopes, and then structure-based identification of epitopes, and the conclusion. So these are the 5 topics which I'm going to show now. So now coronavirus, a few facts. Now let us see. So the dense population, as we're all familiar, is there in Asia, is there in Africa. Not only the dense population, and the lifestyle of this population is also varying compared to European population, compared to us, this American population, because our lifestyle goes close with the nature, wherein we are with the wild animals, we are feeding on the wild animals, we are moving with the wild animals, we are taming the wild animals. So that there is a very possibility that the pathogens from the wild animals are getting transferred to human beings. So like that, here in Africa, there are viral infections like Zika, yellow fever, Rift Valley fever, chikungunya, Ebola, HIV, and in East Asia, the Japanese encephalitis virus, dengue, and all those things. So as I was indicating to you that coronavirus is not new to biological system. That's what the SARS, severe acute respiratory syndrome virus that originated from the East Asia, is a coronavirus. And like MERS, Middle East respiratory syndrome, this is also a coronavirus. Okay. Now from these two also created havoc. That is, in 2002, SARS has come into existence, and MERS came into existence somewhere in 2012. Now, novel coronavirus has come into existence in 2019. And as I was mentioning to you, that we are moving with the living organisms like wild animals. We are feeding them, we are taming them, we are making them for several of our purposes. As a result, this presumption, the presumption is the novel coronavirus shifted from bat to pangolin anteaters, from pangolins, it has come to the human beings because it has been slaughtered in the slaughterhouse. Okay, so that is the reason like this it got spread. So this is the coronavirus. This coronavirus is having the spike proteins all over. So because of the spikes, it has been identified that it is corona. So okay, it's there is a spike glycoprotein, there is a membrane protein, there is a nucleoprotein, there is an envelope protein. So there is a positive strand RNA here also. Okay, still like this, there are 11 to 16 proteins that are available in the coronavirus. Okay, now human coronavirus types. So now I listed just 7 here. So common infection causing cold. Common infection causing cold. There are 4 types of that is coronaviruses, which are shown here. That is these 2 alpha coronaviruses and these 2 beta coronaviruses causes cold. General, like any other influenza infections, here also it causes cold. The remaining 3, as it is indicated in the previous slide, MERS, SARS, and NCO2, they are causing severe respiratory distress. That's what we are now worrying to prepare a vaccine for this. Okay, so why vaccine? Why not drugs? Drugs may cure. Vaccine is a prophylactic device. So as a result, we'll be inducing herd immunity in the population so that the population will never come across this type of the pandemic. Okay, that is the purpose to have the vaccine. Okay, now, so all over the world, wherever the coronavirus erupted, and the samples have been collected by this GISAID, that is Global Initiative on Sharing all Influenza Data. It is based in Germany. And they nearly 45,000 genomes of coronavirus have been collected, novel coronavirus have been collected, and they classified those genomes into several of the clades, like the S clade, B clade, G clade, GR clade. I'll show you clearly in the next slide. So like this, these are all several clades. 39,000 full genomes have been collected as on 12th June. Okay, so there are S clade, 3,000, L clade, so many, V clade, GR clade. So our Indian population also having this clades, this G clade, like this. So if one person is infected with one clade, second infection comes means it must have been due to a different clade. So that's what happening now. So we had to work out vaccines in that fashion. Okay, now a few biological aspects of this coronavirus. So as it is indicated in the previous slide, in Wuhan, coronavirus arose in 2019, Middle East is 2012 in Saudi Arabia, and SARS in 2002, and common cold caused coronavirus like 229E, NL63, OCE4, and HKU, Hong Kong University 1. So these are all, this has been identified in 1960s. Okay, they are caused common cold. Okay, now the current status of Wuhan coronavirus is cases are reported in Wuhan, that is in Asia, and now reported US and other countries. Still, it is prevailing. And with regard to the Middle East, it is declining since 2016. It was prominent and prevalent in the peninsula. Now it is declining. Now, with regard to the severe acute respiratory syndrome, it was no new cases were found starting from 2004. It was there for a short gap of 2002 to 2004. Okay, now it is there all around. So these are all the various proteins. Now we are having wonderful, sophisticated laboratory facilities all around the world. As a result, the protein structures of coronavirus have been deposited in the PDB, Protein Data Bank. Like that, all those things have been shown. We are now in this particular lecture for another 20-30 minutes. We are working with this spike glycoprotein only. So this is the protein which is present on the surface of this coronavirus. Okay, now this is again being very much targeted for the coronavirus not to proliferate its proteases. Okay, so this is another thing, another enzyme protein that is very much being used for the drugs, because this is RNA dependent RNA polymerase. That's what RDRP. If this RDRP is inhibited, the coronavirus cannot get multiplied, proliferated, divided. Okay, so that's what the drugs are also focusing on, this RDRP. Okay, now we are focusing on the development of vaccine so that our immune system develop antibodies which will neutralize the coronavirus, so that coronavirus will not proliferate. Okay, so the main risk with all these viruses that have been shown in Africa and also in Asia is crowding. Because of the crowding, we are facing this problem. So my advice to you and other colleagues of us is avoid crowding. Wherever you are, stay alone. So with this brief introduction and a little bit biology of the coronavirus, we'll go for the informatic tools, immunoinformatic tools, which are being used in the study. Okay, so these many informatic tools have been used, a few by me, like immune epitope database and analysis, and others are by our Dr. Abraham, Professor Venkateswarlu, my colleagues. Okay, so NCBI is a search engine from where we can collect the sequences of the proteins. Like this, we have used this NCBI. And PDB, that is protein data bank, where from the spike protein PDB format, we can collect this. And after having taken this too, then we have to evaluate the epitopes. So that is immune epitope database, wherein if you submit this in the FASTA format, the protein data, the protein sequence of this particular spike protein will be getting huge hits of epitopes. 64,000 hits I have received in different ranks. They all have been classified, filtered later. And again, once all this, again we use this, what is called, computational immunological simulation. This is the software, computational immunological simulation. In this also we secure several of these epitopes. Okay, now the next one. So whenever we go for this epitope retrieval from the sequence that we are submitting, and we have to present the HLA of our population. That is the HLA that is predominant in South Indian population and also in Andhra Pradesh population, which is collected from what is called the literature that I presented to this immune epitope database. As a result, in consonance with that, it has given what is called epitopes. Okay, why we require the HLA? Because HLA is the one which is said to be the carrier of these epitopes to the next level. Because whenever the antigen is given to a host means to the human host, antigen is devoured, that is engulfed by the macrophage or dendritic cell, and the dendritic cell processes the antigen and presents the antigen to the T helper cell through HLA molecules. Okay, that means fragments of this antigen has to get anchored. Fragments of this antigen have to get anchored to the MSC molecule. That's what MSC epitope binding we have to evaluate. That's what immune epitope database we have used. So like this vaccine, wherein the protective and that is antigens and sub-antigens, we work with this. And again, this Lig plot and dim plot we have done for protein-protein interaction. And this BCE, that is B cell epitope prediction. And again, toxin prediction, that is whatever the vaccine construct that we are making, ultimately, whether it may be having toxicity in human host or doesn't have the toxicity in the human host, for which also this, once we submit the vaccine construct, it gives that information. And again, the vaccine construct, does it follow this Ramchandran plot, favorable regions of the residues, and alert of whether the vaccine construct that we make, vaccine construct means nothing but an antigen, it's a protein only. So that is several of these epitopes linked together by certain linkers. Linkers are AAY, CPG, PG. Okay, AAY means alanine, tyrosine, and CPG means this GPG means glycine, proline. So these are the linkers that we use to connect one epitope to other epitope in this, particularly the SNAP gene. SNAP gene is one of the awesome tool wherein we'll be across varieties of the plasmids where you can culture, where you can cultivate the vaccine construct. So once you have the vaccine construct, that vaccine construct can be converted into DNA, a complementary DNA sequence in this JCAP. So that complementary DNA sequence we can construct in this plasmid. We have taken here P828, and in this plasmid, we can, all the plasmid resources are available in the SNAP gene. Okay, there is that is the protein solubility. This is again web-based tool, whether this vaccine construct that we made, whether it is soluble in the cytoplasm or not, so that it will let. Apart from that, the PSA PRED, this is protein structure analysis prediction. Again, it is a blast-based tool wherein the 3D structure will be known. Apart from that, this ITASR, that is iterative threading assembly refinement. So in this also we'll evaluate the 3D structure and function of the vaccine construct. So many things we worked, and ultimately we come to the conclusion. That's what I'll be showing you now. So now with this informatic tools, we proceed for the rest of the two sequence-based identification of epitopes. So again here, sequence-based and structure-based, both the things we are focusing. So now, what is the aim of our study here? To predict peptides from spike protein, S protein of novel coronavirus 2, to bind with high affinity to HLA class 1 and 2, T cell, B cell, to design vaccine. See, so many pictures, so many intermediaries are coming into the picture, like HLA class 1. Without HLA, we can't start the immunological work because it is the one which presents the epitopes or antigen fragments to the T cell, data to the B cell. Okay. So the B cell is the one which produces the immunoglobulins. These immunoglobulins neutralize this particular pathogen, even if you are getting infected in the later cases. Now, just the hypothesis testing for those beginners in the research and all those students, whenever we propose anything, we have to develop a hypothesis. Okay, here null hypothesis and alternate hypothesis. So predicted peptides, that is all through bioinformatics, whatever we do, it is only prediction. The prediction will be correct if it goes through in vitro, in vivo analysis and in situ observations. Okay, now, so null hypothesis here is predicted peptides from spike protein, S protein of a novel CoV2 will not show in silico binding with high affinity to HLA class 1, 2, T and B cells, so as not to design vaccine. That means it's a negative approach. That means our study will not work. That's what null hypothesis says. Alternative hypothesis, predicted epitopes from S protein of nCoV2 will show in silico binding with high affinity to HLA class 1 and 2, T and B cells, so as to design vaccine. That's what our ultimate game. If at all our results come, we say that alternative hypothesis we accept it. Okay, thank you. With this, we proceed the work. Now, as I was telling to you, there are several softwares. All those softwares are built based upon the concepts that have been developed in immunology and molecular biology. Okay, so they are not standalone, made in collaboration with our biological scientists. Now, a few of the scientists I will mention, still there are plenty, and the Ilya Marchenko was the first Nobel laureate in immunology, and somewhere in 1903 so, and he has shown the phagocytosis. And again, and he also explained about the cellular theory of immunity. Then the second one, Zinkernagel and Doherty, their work is very prominent wherein T cell receptor complex, there's a TCR plus MHC plus antigen present. MHC means major histocompatibility complex, antigen presentation. Then Bruce A. Bulter and Jules A. Hoffman, they did work in relation to toll-like receptors. Toll-like receptors are the first sentinels to identify the pathogens. Okay. Then Benny Seraph and Dossett and George Snell, they did work with molecules, particularly in transplantation of organs. Okay. Then R.M. Steen, they worked with dendritic cells. Dendritic cells are the first cells, immune cells, which recognize the pathogen and process this. And again, Niles Jerne, they did work with regard to the lymphocyte clonality. Apart from that, these immunoglobulins have been done by Porter and Edelman. They identified the structure. So the Nobel laureate lectures series helped us in identifying what softwares and those concepts have been here. So I will let you know a few of these things here. Let us say this is a dendritic cell. Dendritic cell on the surface is having TLRs, toll-like receptors. With these toll-like receptors, it identifies the antigens. And those antigens are endocytosed. Once the antigens are endocytosed, they are processed. The processed antigens are presented through this, what is called HLA molecules. And this processed antigen, see, this is the dot here. This is anchored. This anchored to the anchored to HLA molecule. That means this is the this is the epitope of the antigen anchored to this. And this is further being conveyed, presented to the T helper cell. To the T helper cell is having T cell receptor complex. T cell receptor complex. It gets anchored here. And again, that means T cell receptor epitopes. Then again, this T helper cell presents to the B cell, again, B cell receptor epitopes. Epitopes here. Again, okay. And this B cell produces immunoglobulins. These immunoglobulins neutralizes this antigen. So this is the total concept. So on this concept, the softwares have been made, and those softwares we are utilizing now to develop multi-epitope vaccine, okay, for this coronavirus. Clear? Let me go to the next slide. Okay. Now the spike proteins and diagrammatic representation of epitopes of a folded protein. So this is the coronavirus. This is the spike protein from PDB, the protein database, and for those of the youngsters, so the epitope means something protected. So there is another. This is also a portion of the protein that is hidden. This hidden portion cannot be seen by the cells like dendritic cells and all those things. So any protein may be having several epitopes. Here in this case, for the diagrammatic purpose, I've shown for the students that there are 24 epitopes which can be perceived by an immune cell. Okay. So advantages of epitope vaccine. So now there are vaccines, attenuated vaccines, I told you, heat-killed vaccines, I told you, recombinant vaccines, hepatitis B surface vaccine, which is there in the market. I told you all these things. So in that, there is advantages of epitope vaccines, more specific immunity. That is, we are focusing only on the epitope in this particular vaccine. So that means in this vaccine, we take only this epitope, not the hidden portion. As a result, preparing the vaccine is also commercially easy. And again, our system will also see only this portion. Okay, now our divide of side effects in contrast to entire viral protein vaccines, the ability to deliver high doses of potential immunogens at a lower cost. Okay, and these epitopes are having conservancy all over the world, and they are stable binding with HLA class 1 and class 2. HLA class 1, any one of you wanted to start to devise a vaccine through in silico means HLA of your area. You have to collect from the literature and evaluate the epitope binding. Okay, and again smooth passage through TAP. TAP means transporter aided protein, which is present on the endoplasmic reticulum, where from this HLA class 1, HLA class 2 are getting transferred to the cell membrane. Okay, that's what now sequence-based, structure-based, the structure-based molecular docking calculations, the sequence-based binding properties to MSC molecules. Okay, the workflow. Selection workflow of our anyone, if we want to start this type of what is it called, multi-epitopes to design the vaccine construct. To begin with, we had to select an immunogenic surface protein. Surface protein is most important here because hidden proteins are not being seen by the neutralizing antibodies. Okay, so this needs to be presented, and then once it is presented, the protein may be around 1500 amino acids, 1000 amino acids. So the computer system may not know. It will select all polar amino acids as epitopes. Polar amino acids means glutamic acid, aspartic acid, and tyrosine, and what next? Lysine, and arginine. They're all having the charge. So with wherever the sequence they are found the charge, it projects the combination of the epitopes. So in that I have got now 64,000 hits. With all that we cannot work, so they had to be filtered. Like let a screening of HLA haplotypes of the population, our haplotype, our region, wherever you are there. And again, optimization of the complexes by docking calculations and immunogenicity evaluation, particularly CIM, that is what is it called, software, that is computational immunological simulation software. It gives enormous what is it called, the predictions connected to the immune responses. That is an excellent software program. Okay, sequence-based approach. Let me tell you. So the spike proteins of SARS, bad SARS, and SARS of that is from China, and MERS, and again the novel coronavirus from Wuhan, and the NL3 of alpha, and 229 of alpha. So they're all approximately between 1100 to 1300 amino acid sequence. Okay. Now, the length of the this spike protein is divided into two domains. One is S1 domain, the next one is S2 domain. Okay, S2 domain is the one which is having the spike. Okay, and not only that, this S2 domain is the one which makes the virus to have fusion with alveolar epithelial cells, so that this fusion make it to get endocytosed inside. Now, like S1 domain is having what is it called, receptor binding domain. The receptor binding domain ranges from 300 to 500 here, and in this in our novel coronavirus, it also varies between 319 to 541. Okay, so much of the epitopes we focused here in this region. So this is the region where if it is blocked, then the coronavirus cannot anchor to SCE2 so that it cannot get endocytosed. So that's what our plan is. Okay, so whenever we submit the protein sequence of 1273 protein sequences, then the what is it called? The software gives as many possible, so many epitopes. But we wanted this only in this region. So like that, we have done the filtering and we got the values. I will let you know in the other slide. Okay. Now, percent identity matrix. So if you work out among the 6 what is it called coronaviruses we selected, the Wuhan coronavirus is more close to the SARS and the BAT coronaviruses, and it is least close to this what is called the remaining 2 alpha coronavirus, 27%. So that's also we require, because conservancy analysis we had to do in this process. It helps. So now epitopes identified within the receptor binding domain. That's what I was mentioning to you. That is what based upon this HLA and HLA-A. So now, as I was indicating to you, IEDB, Immune Epitope Database, we presented this spike protein sequence, and in that we are interested to get to know about the epitopes for the HLA haplotypes. The HLA haplotypes are shown here, and so many peptides we have resulted, and they're all ranked like this. Just I'm showing you the raw data in few slides. Like this again here, this all the various ranks that we have received. Slide. This is what the filtered one. This is what the filtered data of 64,000 hits. Now, please know the HLA haplotypes of the population of Andhra Pradesh. They are having, that is HLA class 1, HLA class 2. Within the HLA class 1, class A, class B are highly polymorphic. C is least polymorphic. There is E, F, G are there. So one important news I wish to let you know here is, see, when a fetus is there in the uterus of the mother, the fetus is not same as that of the mother with regard to the HLA. The fetus is half mother, half father with regard to the HLA, but still mother bears that heterozygous, what is it called, fetus, because the fetus never have this type of HLA haplotypes expressed. It will express only HLA-Z, HLA-A, B, C, E, F, G. G is least polymorphic. That G least polymorphic HLA of the fetus is being accepted, tolerated by the mother. That is the reason why the fetus is maintaining the conceiving stage. Okay, so that is another important thing, a different aspect. Now let me know about the HLA of Andhra Pradesh. A1, B57, DRB1, 7. And A33, B44, DRB107. A02, B40, DRB115. And A24, B07, DRB115. A24, B40, DRB115. So like this, haplotypes are available in Andhra Pradesh population. So haplotypes means this particular sequence will get inherited as it is without crossing over or linkage during meiosis process of the germ cells. Okay, that's what haplotype. Okay, let's now see. So the epitope, as I told you earlier, receptor binding domain. So we have selected the epitopes only in the receptor binding domain, start sequence, end sequence, and we have selected the high rank epitopes. Okay, so like this, we selected 10 here, 10 sequences. Okay, I'll go to the next slide. Okay, so those sequences again we submitted to cluster analysis. So if these sequences show some identity, they'll be given a sort of what lines. These two peptides are linked by a line. So that means they are shown two peptides like this. That means all these peptides which we selected, they're all having identity features in developing immunity, in eliciting immune response in the host. Okay, so that's what about the sequence based. Now structure based identification of epitopes. So this much of this work was done by Dr. Abraham. Now, so here in this particular case, it is done by ITAS, that is iterative threading assembly refinement. Okay, in this the chain A is TLR, toll-like receptor, and the chain B is the vaccine construct. So whenever we give a vaccine construct to an individual, okay, so vaccine does not directly go to B cell. To begin with, it is being received by a dendritic cell or a macrophage. So those dendritic cells are having TLRs, toll-like receptors. Those TLRs, protein-protein interaction. That's what TLR is a protein, vaccine construct is a protein. Both PPI, there is protein-protein interaction of this vaccine construct with the TLR is shown here. Okay, so that's what generated, and it is interacting. I'm going to the next slide now. Yeah. So once the vaccine construct is made, whether this vaccine construct is only random coils, is only beta pleated sheet, or is also having with it called alpha helix, or right-handed alpha helix. So that's what we submitted to CyPED, that is protein structure analysis through prediction. And in this, there is a helix. Helix is the pink one, and this is what is this called random coil. This is what called the sheet, beta pleated sheet. But in the next, this is the next one is protein solubility. So the vaccine construct, whatever we made, that's submitted to the protein solubility software, wherein our query construct is almost equal to the standard that is being shown in this protein solubility. That is, our vaccine is soluble. Okay, so in our serum there are a few soluble proteins. Insoluble proteins are also there, but it is soluble. Okay, that's what we want a vaccine to be soluble in cytoplasm. So now this is a wonderful diagram, Ramchandran plot. G.N. Ramchandran, who was there in Madras University, later shifted to ISC. Now he did this enormous work somewhere in 1950s, 1960s, where there was no computers at that time, very popular. All over the world, wherever you see the biochemistry books, bioinformatics books, G.N. Ramchandran plot is being shown. Without that, there's no replacement for this. Okay, so here, this is Ramchandran plot wherein alpha helix, all the construct the residues which are present, the residues which are present in the vaccine construct, there a few of them are alpha helix, right-handed helix, a few of them are beta pleated sheet. Everyone is within the favorable region, in the favorable region of this zone. Otherwise, they are not away. They are not away from this favorable region. Okay, and again he can say this is the phi and this is the psi. High value. They're all between 60 and 140. That's all detailed so as we study regularly. Okay. So now, with regard to the allergenicity, so for which the vaccine construct allergen, when after having submitted to AlertTop, it has given probable non-allergen. The construct is non-allergen that has been identified by this. Okay, so this is what I was mentioning to you. One of the wonderful, what is that called, the software tool, you all have to practice SnapGene. So in this SnapGene, there are plenty of resources about the plasmids. So we have taken PET28 plus plasmid. So if in this plasmid, this particular region, violet region, is the one where we have kept the DNA of our vaccine construct. vaccine construct DNA is converted into cDNA by cat. That that cDNA has been pasted here, and this has given the results like this. We can also regularly in vitro studies, if we want to develop a vaccine construct, we can get out of this from many bacteria by inserting this plasmid. Okay. That's what the advantage. It is an awesome program. I'm going to the next slide now. Okay, so now this is the ultimate simulation. That is, now we have done the vaccine construct through computer tools. Okay, whether this vaccine construct is having the capacity to elicit immune response. Does it elicit immune response or not? That also we can do the simulation studies by using computerized emulation, computerized immunity simulation software server. Okay, so the CIMSIM, if you take up this set of the server, then we can work continuously for the next 3 to 4 slides. Okay, we'll be ending with another 4 to 5 slides. Okay, now once you submit the vaccine construct, to begin with, T helper cells, are they responding? So the T helper cells are active. Active T helper cells are this. So active T helper cells are so many are representing here. So this help the T helper cells will be there active up to 100 days. That is from 0 day of infection up to 100 days. So we'll not get infection for another 3 months. That's one. And again, duplicating. So the cells are replicating because they are recruiting. The T cells are more recruited upon infection. That's what that's also been shown in this simulation studies. Okay. The next one is possible B memory cells. Okay. So the B memory cells, this is the color. The B memory cells are also increasing. That is, B memory cells means are nothing but the plasma cells. The plasma cells, that's what nowadays people say, plasma therapy. Plasma therapy. That means they take these plasma cells, and the plasma cells produce these immunoglobulins and give to the patients who are suffering, so that the immunoglobulins will neutralize the novel coronavirus. That's what the plasma therapy. Okay, now this B memory cells are also increasing starting from the 50th day, and they maintain almost up to 250 day. Memory cells are more in number. So these are simulation studies. It's not the in vivo studies. Okay, even the simulations, after having submitted the vaccine construct, it gives the prediction. It gives the prediction. Clear? Now, IgM, IgG levels using a computerized immunology simulation server. Okay, here IgM. Now, all over the world, people are doing the rapid blood test. Rapid blood test. The rapid blood test means IgM, IgG, they are present in the serum, they are being evaluated. If you are having IgM followed by IgG at a appropriate level, people will suspect that we are infected. That's what IgM levels and IgG levels. So IgG levels are also, this is the IgG levels, Ig1, Ig2. These are the Ig1, Ig2. So they are also maintained up to the level of 150 days. Okay, so IgM, IgG is also shown as a simulation aspect here. Now, this is the last one. Here we have to focus much. See, this CIMSIM server not only showing the immune response with regard to the T helper cell hike, B memory cell hike, and here it is showing the interleukin storm. So whenever we have got such a type of the infection, several of the cytokines and interleukins are the ones which communicate cell to cell. So like this server predicts the production of these interleukins. So in that, please see one of the important observation that the interferon gamma, I think so it is visible to you, interferon gamma levels are high. Interferon gamma levels are high. That means interferon gamma is antiviral. So this construct what we have made is working very well, and it, given the response of the production of the interferon gamma, antiviral cytokine. That's what the interferon gamma is, even up to the level of 100, it is high. So this cytokine, if it is high, means it promotes cytotoxic T lymphocytes to produce more, to recruit more cells. Apart from that, IL-2 IL-2 is interleukin-2. This is again hike apart from that is upon vaccine construct submitted. So this IL-2, the major function is recruitment of immune cells. That is, whenever there is a defense requirement, is you recruit more number of cells. How do they get recruited? For that, there is a cytokine. That is what IL-2 promotes the number of cells more. Okay. So all these simulations. Yeah, please, ma'am. Please, ma'am. So this is all the various raw
Yes.
data that we have. So these are all the various raw data that we procured, with which we may not be in a position to have this. So here, this was the prediction of the toxicity. There's no toxicity that we have got out of this software toxin PET server, and helper T cell peptide epitopes are also identified here. And ultimately, we have come to the last slide conclusion. Novel coronavirus 2 is still enhancing its presence, developing into several clades. Sequence-based identification of epitopes at receptor binding domain of S protein resulted in 10 epitope sequences with a maximum identity of 80% and bound to HL haplotypes of the population of Andhra Pradesh, India. Structure-based identification of epitopes bound to HLA led to the construction of vaccine vector and evaluated secondary structure, allergenicity, elicitation response, upsurge in IgM and IgG levels, B memory cells, and cytokine storm, interferon gamma inducing ability, and IL-2 bailout. All these things we observed through this CIM SIM servers, and null hypothesis rejected, alternative hypothesis accepted. So that is our work. And ultimately, immune boosting is the only solution for us to get protected from frequent viral eruptions, particularly Tulsi, Amla, nowadays Stanospora cordifolia, and neem, all those things will be of some use. Thank you. These are our publications, recent publications. Thank you all. Thank you particularly to Divya Sri Madam for spending whole of her time only on this webinar. Thank you, Madam.
Thank you, sir, for the wonderful talk. So participants, you may post your questions. So there are some questions, sir. I would like to read out one by one, and also I would actually switch on for some of them, switch on their audio for some of them. So yes. So there's a question from Godwin. So he's asking, do you plan to extend your in silico experiments to in vivo experiments in future?
Yes, yes, madam. There is possibility because here one advantage of in vitro and in vivo are there, because the plasmid we identified here through in silico means P8 PET28, that is available commercial. You can buy that PET28 and insert the rDNA sequence of what we propose here, and with that we can produce what is called total vaccine construct. Okay, so that vaccine construct, once you can give to to some of our people, local people, or again the mouse and all those things. Let us see, after having been exposed to this type of the viral infections, whether they will work or not. We can do this type of the work. It's not at all a difficult process. We got the animal house is there with in our university. We're also having the clean room where we are maintaining the cell culture, animal cell culture. It is possible. There is possibility for us to do this work, madam.
Thank you, sir. And there are a couple. I think another question from him. Why did you select a spike protein for your study?
Yeah. So there are about nearly 11 to 16 proteins that have been reported. Good. So in that, what is happening is, I was mentioning to you, whenever any bug goes in our system, okay, the surface of the bug is being seen by other immune cells. The deeper portion of the bug is not seen by our immune cells. So fortunately, with regard to the coronavirus, the surface is only the spike proteins, which are nicely, very well projected out. So that's what we have taken the spike protein. Apart from that, the spike protein is most essential here because the spike is the one which is facilitating the coronavirus to get endocytosed. Okay, so once the spike proteins are neutralized by antibodies, then there is no possibility for this to make it membrane fuse and then endocytosis. That's what we have taken the spike protein. And again, we have taken within the spike protein receptor binding region of the spike protein to develop the epitopes, madam.
Thank you, sir. And another question is, so there's a question from Kanak Chakraborty. What are HLA haplotypes? And another question from him is CIMUSIM server is a paid software?
Yeah. So HLA haplotypes. HLA haplotypes, that is human migration, basically evaluated through HLA haplotypes. That's one thing. Let me tell you about HLA haplotypes for some more time. And the HLA is human leukocyte antigen. Okay, a human leukocyte antigen is considered to be self molecule. That is, the definition that we used to teach to our students in immunology is what is the tolerate self, target self. Tolerate self, target self. What is tolerate? Self. Self means our own MSC molecules we are tolerating. Non-self means that is your MSC molecules are non-self to me. Your MSC molecules are non-self to me. That's what HLA is being spoken at length in immunology. Okay, that's next. About again HLA haplotype. HLA is present. That is the antigen means it is having its corresponding genes. The corresponding genes are present in on the chromosome 6, madam. Chromosome 6. Okay, in this chromosome, HLA1, HLA2 genes are present. HLA1 again, HLA class 1 contains A, class A, class 1A, class 1B, class 1C, class 1E, class 1G. So these are all said to be HLA class 1. HLA class 2 means DP, DQ, DR, DM. Okay, so this 4 comes under this category of HLA class 2. They're all present together on what is this called chromosome 6. Okay, and again haplotype. So haplotypes means I may not have all the combination of these HLAs. Okay, now within the HLA class 1 there are 1,000 alleles. Within the class B there are 600 alleles. That means your allele, my allele will vary. Okay, so like this, all these alleles what I'm having, they are present as one cassette, in that particular chromosome. Okay, so what is happening is with regard to the gametogenesis time, germ cell formation time, there is a very possibility that crossing over linkage occurs. As a result, the haplotype may get disturbed, but that's not so. Get disturbed is not called haplotype. So this haplotype combination get inherited as it is to the offspring. So that's what it's called as the haplotype, HLA haplotype. Now, with regard to the CIMSIM, one of my friend, Dr. Abraham, has worked it, and I do not know whether it's paid or free, and I will speak to you. I'm not sure. Okay, next, madam.
Next question is, coronavirus and SARS virus are of same class, isn't it? So what are the main differences that they hold?
Yes,
and and if a person affected with a particular clade of virus that it belongs to and recovered, what is his possibility to be infected by the same clade or any other clade again? So this is a question from Sujata Nair.
Yeah. So, yes, madam. So we have done the phylogeny of this coronaviruses. Particularly, I selected only seven or six varieties coronaviruses, and out of the what is that called, the genome sequences that are available in NCBI. Okay. Now, out of that, we found out the bat coronavirus and SARS coronavirus constituted one primary cluster with a good bootstrap value. And with this primary cluster, there's an offshoot called as the NCOV2, that is our novel coronavirus. Okay, so with this, what I wish to let you know is, the novel coronavirus and SARS coronavirus are very closely related. And not only they both related, they're also related to the bat coronavirus. Now let's come to the point. So now another important feature of between these two is coronavirus. Novel coronavirus and SARS says both of them are having, this receptor binding domain that will anchor to that is angiotensin converting enzyme 2 enzyme. Both of them will anchor to the same site, So in that respect also, these two are same. Okay. Now with regard to the infection, so this
Yes, sir.
coronavirus, if it is infected, if I get infected with one clade, after some time I may get infected with some other clade. That time it may not be as intense as that of the first infection. It may not be as intense as that of the first infection. So that's what, if we are having the immunity, that is herd immunity, that's what, if you are primed with this type of the coronavirus spike antigens, we may not have the infection. So did I answer your question, madam?
Yes, sir. Yes, yes.
Thank
So there's a question, sir. So the couple, we can deal with another 4 to 5 questions,
Yes,
and
madam.
maybe stop with that. So
Yeah.
there's another question. Will single epitope based vaccine designing be good enough for COVID-19, especially when it is reported that spike protein is mutating? So this is a question from Rajan Chowdhury. So will single epitope based vaccine is good enough for COVID-19?
Yeah. So, this is a good question, that spike protein is also undergoing mutation, and so far we are coming across synonymous mutations, madam. Synonymous mutations. As a result, the amino acid sequence is not varying. As a result, the amino acid sequence is not varying. So, as a result, the this receptor binding domain will be having the same thing. There's a very chance that the spike protein mutates. If the spike protein mutates, means there is it may not become a pathogen to us, because it may not bind to acetylcholine. There's what is the angiotensin converting enzyme. Okay, so now let us see. As far as now, it is undergoing synonymous mutations in the short duration of time. That's what this GCI, that is Global Initiative on Sharing, that is influenza data, they are showing. Okay, and single epitope, what you are asking, sir, the single epitope vaccine may not be recommendable. Because multi-epitope, as I have shown you in one of the diagram, that 24 epitopes I have shown. That is, there's a very possibility for the immune cells not to miss this virus, because there are several immunoglobulins. Anyone can capture it. Once it is captured, means immune complex will form. Once immune complex form, complement system will come into the picture. That's what the single epitope is not recommended. Always multi-epitope vaccine, because no, epitope is only 9 to 14 residues, sir. If it is an antigen, means that is hepatitis B surface antigen, which is nearly 1,000 amino acids, But the total vaccine construct that you are giving will not be beyond around 500 or 400. What is this called, residue? So single epitope is not recommended. Multi-epitope, we have to, that works now. Okay.
Thank you, sir. Thank you. And another question: how coronavirus jumped from animal to human in terms of species specificity?
Yeah, good. Species specificity. So here, host pathogen species specificity is very well established, and the one which is there, the animals should not come to us. But what is happening is anthrax. You are all familiar with the anthrax. Anthrax is quite there in all some of these what is slaughter animals. The slaughter fellow will get this anthrax infection, because he is very well involved in handling that slaughtering business. So like that, it jumps into the human beings also. There is a different issue. Now with regard to the coronavirus, what origin can be traced from the bat. From the bat, it came to what is it called, anteater, and from the anteaters are being sold in the market. And in the process of selling this, the precautions that are taken must not have been very cautious. As a result, it must have infected a few individuals somewhere in the December or October. and gradually that fellow who got infected must, because one of the other important feature is the SARS coronavirus, BAT coronavirus, and novel coronavirus, all three are having unfortunately one that is receptor binding domain. That receptor binding domain is binding only to acetylcholine, that's angiotensin converting enzyme, 2 angiotensin converting enzyme 2. If it is not specific to angiotensin converting enzyme, we must not have got this type of infection in humans. So that is the main reason that it caused the host from the what is it called, bat to this anteater to our humans. Okay, so that's what it has caused the host.
Thank you, sir. Thank you. And there is another question. What is your opinion on plasma therapy? So this is a question from the head of bioinformatics, CPMB, Dr. Nyanam ma'am. So, and she would be talking soon as well. So she has a question. What is your opinion on plasma therapy?
Yes, ma'am. Thank you, ma'am. So this is the therapy. Plasma means it's not the serum, plasma, and blood cells and the blood. Plasma means it's a plasma cell. Plasma cell is the one it produces immunoglobulins. So that's what here the plasma therapy, the term plasma therapy is not new. The earlier days also, they used to give the blood of the recovered person to the infected, person. Now let me elaborate on this plasma therapy, madam. So the person who got infected with the coronavirus and recovered from coronavirus, as it has been shown in CIMSIM, what is the software, it persists. The what is it called, immunoglobulins, IgG persists, even helper T cell persists, even memory B cells persist till 150 days. 150 days, almost 2 to 3, more than 3 to 4 months in the serum. Okay, so as a result, if we take the blood from the recovered person, recovered person, the blood contains plasma cells. The plasma cells are the one which produce the immunoglobulins. They are the progeny of the B cells, and those cells, once it is given to the patient, and in the patient, the plasma cells will produce good many number of immunoglobulins against this coronavirus. So it will, what is that called, neutralize the coronavirus infection. As a result, the intensity of the infection gets reduced. Plasma therapy is now being practiced, madam, and for which the recovered patient has to voluntarily give the blood, and from the blood we should have the capacity, the infrastructure to isolate the serum and the plasma cells, and we are not giving blood as it is to the patient, and we are filtering only the plasma cells, and those things are being given along with the serum. So it works, madam. It certainly works.
So, what is the compatibility of plasma cells from donor to recipient? So, in that note, there is another question from Rajan Chowdhury, and so he's asking, what is the compatibility of these cells from donor to recipient?
Yes, ma'am. It is a very interesting question, madam. Compatibility. So, whenever we speak of the compatibility, the HLA molecules speak. But here, that's what I'm telling to you. Once we take the blood from the recovered person, the whole of the blood we are not injecting into the patient, because the whole of the blood contains RBC, contains WBC, contains several things contain. So out of that we filter, we provide the serum. Okay, the serum does not have the HLA molecules. So there is no problem of compatibility. Another problem is the serum may contain what is called A blood, anti-A or anti-B, some of those things. Those things can be worked out, whether it is having anti-A, anti-B. If it is anti-A means we should not give to the A blood group person. If it is anti-B means B blood group person should not give, madam. That is another important thing we have to take into consideration. Or otherwise RBCs of the patient will get clumped. That's another thing. So with regard to the compatibility of plasma cells, if we take up, plasma cells longevity is only 3 to 4 days, madam. It reproduces, longevity is only 3 to 4 days, and the plasma cell produces immunoglobulins. Those immunoglobulins will cure for the neutralization. So the compatibility problem with regard to the plasma cells will not come. only with regard to the serum anti-A, that means our blood groups are A blood group, B blood group are there now, AB blood group. So wherein the anti-A, anti-B will be which has to be evaluated.
Thank you, sir. And so there's another question. So epitopes of coronavirus causes any pathogenesis in human?
Yeah. Epitopes are only part, very small part of the protein. They do not cause any pathogenicity. Pathogenicity as a whole protein causes. That is, spike protein as it is causing pathogenicity because spike protein is making the virus to penetrate into our system. So that is the pathogenicity. In that spike protein, epitope is one. Okay, as it is, epitope is not standalone. Epitope is not standalone. Epitope, along with the spike protein, is a part of it. So, epitope will be useful for us for the investigations and for the preparation of multi-epitope vaccines. Okay, epitope is called, if at all you want to say in the normal terminology, it is antigen determinant site. We're having a spike protein. In that spike protein, how many antigen determining sites are there means they are nothing but the epitope. Okay, epitope as it is doesn't cause the pathogenicity. Protein will cause the pathogenicity, as far as my knowledge goes. Thank you.
Thank you, sir. And there's another question that's regarding, again, following that plasma therapy question. So we know that every cell are specific from person to person, including plasma cells. So will it not activate the immune response? So this is another question again from Rajan Chaudhary.
Yes. Plasma cell, that is my cells and your cells. If you take up, my cell contain one type of HLA, your cells contain another type of HLA. That's what I told you in immunology. The definition that you give in the class is, the definition for immunology is tolerate self, target non-self. So that means that's what madam is asking about the compatibility. The plasma cells longevity is less, madam. Okay, so that is the advantage of avoiding the what is called the compatibility, and that is not a risk because the anti-A, anti-B, that is blood group A, blood group B. Blood group A is having anti-B, blood group B is having anti-A in that serum. Okay, if that anti-A is there, means if the patient is having A blood group, this it causes menace because anti-A agglutinates all RBCs. That is the problem. Okay, so that aspect of the serum we have to take into consideration. And while giving the plasma cells may differ from the patient. If I am a recovered patient, I am now giving my blood plasma cells to the patient, and patient HLA, my HLA will be varying. Madam, patient HLA, my HLA will be varying. But the longevity of whatever the cell that I am giving as a host, I am as a donor, longevity of the cells of plasma cells that I am giving is very short. Plasma cells survive only for 4 to 5 days or 1 week. Okay, so that type of the compatibility will have, but though it survives for a week time, it proliferates. There is a possibility that plasma cell continuously proliferating. Memory cells. Okay, madam. So in that short span of 7 days of its survival, it gives whatever bails out, not only gives, bails out immunoglobulins. Those immunoglobulins neutralize the what is called the patient coronavirus. So that 7 days, the patient recovers. Okay, so this plasma cells will not be there in the patient forever. It is only for a duration. So that compatibility problem doesn't exist here, madam, regarding the plasma therapy.
Thank you, sir. And there is another question. Can we use epitopes itself for immunosim analysis? Because sometimes we plan to use epitopes as different form of vaccine.
Yeah. Now we we feed protein sequence. We feed protein sequence into CIM, madam. And CIM gives us not only the protein sequence, we give the HLA haplotypes also. Our HLA haplotypes. Okay, madam. So once the faster sequence of this gene construct is given along with the HLA haplotypes, we get the epitopes. Those epitopes, and again several of the simulations of immune responses we evaluate, madam.
Okay, sir. Thank you. And there's a question from Ratan Das: how to avoid storm? Yeah,
How to avoid?
cytokine storm.
Oh, cytokine storm. Okay, so cytokine storm is the one. There's no need to avoid the cytokine storm here, because once we are inflamed, good many number of cytokines play. They come and play. They play in such a way that they communicate. They communicate to the, what is it called, other immune competent cells, immune accessory cells, to act against the pathogen. So as a result, the cytokine storm in this case, the interferon gamma, we have seen in this diagram, interferon gamma took a high peak, what is it called, eruption of the release. So this interferon gamma is antiviral. So with regard to the, once the interferon gamma is more, we'll be having more of cytotoxic T lymphocytes. Cytotoxic T lymphocyte is the one which is antiviral. Okay, so that's the reason why cytokine storm comes, because of this coronavirus. And in this storm, interferon gamma is very much acting in favor of the patient, madam. And again, IL-2 also is getting released. That also is working in favor of building the defense. In the patient, madam. IL-2 is the one which recruits more number of immune accessory cells and immune competent cells in the patient body, madam. The storm is very much essential for the patient to build his immune response, madam.
Thank you, sir. And I will go two more questions and we will wind up, sir. And participants, you may post the questions to us, and you may write it in your feedback forms as well. And with that, we will, with one more question, we will stop. So there's another question from Dr. Nyanam, ma'am, head of bioinformatics from TNAU. If vaccine is developed, how many doses at what interval?
Yeah. So it is vaccine. Once the vaccine comes, now we are all doing the simulation studies, madam. Vaccine is developed as usual. It is in a frequency. The frequency is month after month. The first initial dose, later secondary dose, tertiary dose, final dose. But first 3 months we can give. Later, if we give first 3 months, later 1 year. Later 1 year, if we give, that's enough for building resistance against the coronavirus. That's what the hepatitis B surface antigen, which is being provided earlier by Biotech. We have taken first month, second month, third month, and later at the end of the year. So we are free from hepatitis B surface antigen. I advise every one of the people around us now in the webinar, you also promote among your people, hepatitis B surface antigen vaccine has to be taken by every one of us, particularly the before marriage, because we do not know who is having hepatitis B virus. If a husband is having, it goes unnoticed. It goes unnoticed. As a result, the wife gets, the child also will get. In the process, it leads to the, what is this called, liver cirrhosis or liver cancer like this. So you can give at an interval of 1 month, at an interval of 1 month, and at the end of an year.
Thank you, sir. And I think there's a few more questions, but it's already running late. And then we will just conclude the session with vote of thanks. So we have Dr. Nyana ma'am to deliver the vote of thanks. Ma'am, kindly unmute yourself for providing the vote of thanks.
Thank you, sir. I am here to propose a formal vote of thanks to Professor S. Krupanidhi, Head Department of Biotechnology and Dean Planning and Monitoring, Vignan Research Foundation, Guntur. Definitely, it's a very excellent and exhaustive lecture. And I really appreciate the following points during his lecturing. First is about the immunoinformatics tools, what he has given to the participants. Very nicely he has explained like how to use this and where it comes and all those things. And the second one is the flow chart, what he has explained. I think in total, in one slide, whatever he has presented very nicely in the beginning itself, he has explained. And he was also insisting about the knowledge on the different kind of clades and the HLA types before you start working on this, which is really appreciable, sir. And another one is he was also talking about the sequence based, structural based, and the importance, the toxicity studies. Almost, I think all the points, whatever in silico he could explain very nicely. He has explained, and definitely, I feel it is going to be it is really useful for the participants, and that will give a very good idea for the people who are going to work. Then I would also like to extend my thanks to Dr. Divyashree and Dr. Magesh, who is always running to arrange this kind of webinars for the benefit of the students. I totally appreciate them. And then on part of TNA, I also thank our director for permitting us to conduct this kind of continuous webinars for the benefit of the participants. Thank you. Thank you for the opportunity.
So participants kindly, thank you ma'am, and participants kindly post your questions to my email ID, and we will forward it to Krupanidhi sir, and then we will forward it back to you. I hope if that is okay, because we are running out of time. Please post your questions to this mail ID. Thank you, sir, and we will wind up the meeting now. Thank you very much for the wonderful talk and for patiently answering all the questions. We still have a few more questions, but it is already very late, so we will wind up the session for now. Thank you very much, sir.